How are we doing, guys? It's Mr. Striz. I'm signing in. Today's LAQ is how can mutations affect phenotypes and genotypes? So we're going to look at a couple of different mutations as well, and then we're also going to look at whether mutations are always harmful, whether some can actually be beneficial, anything like that. But before we begin, I kind of got a quick video. There's actually some different mutations that actually have been beneficial, and we're just going to watch a little bit of that first before we begin our notes here.
But normal age-related skeletal degeneration as well. Number 5, super endurance. Finnish Olympic skiing champion Eero Mäntyranta, along with his extended family, had a mutation which allowed them to carry 50% more oxygen in their bloodstream. This mutation basically gave them super endurance powers. Number 4, super strength. This boy, by the name of Liam, has a rare genetic condition in which his body doesn't produce the protein myostatin. Myostatin inhibits muscle growth, and because he and others lack it, they're able to put on massive amounts of muscle, far easier than anyone else. The only downside to such a condition is you need to eat a lot to support it. Number 3, needs less sleep. After conducting genetic tests on sleep participants, University of California, San Francisco researchers were able to locate a mother and daughter who shared an abnormal copy of a gene known as DEC2. This gene affected the circadian rhythm. The result is they're short sleepers. They need far less sleep than the average person. This genetic disposition may affect as much as 5% of the entire population. Number 2, resistant to HIV and the Black Plague. In the 1300s, the Black Plague ravaged Europe and killed millions of people. However, there were some people that no matter how much contact they had with the infected, they didn't manifest the sickness themselves. No one knew why this was. They assumed God had protected them. Now jumping forward hundreds of years to the 1980s, when the AIDS outbreak was underway, a story comes about of a man who received a blood transfusion infected with the HIV virus. The fact that the man had not contracted HIV astonished doctors, and for many years they researched and came upon a startling discovery. Some people in the world have a genetic mutation in their immune system that protects them from contracting HIV and the bubonic plague. This mutation is extremely rare, and it requires both your mother and father to carry the mutation in order for it to be passed down to you. And your number 1 genetic mutation is hyperphotographic memory. Everyone's heard of the term photographic memory. It's where a person can recall what they read or see in extreme detail. The catch to photographic memory is the person must commit things to memory. However, a person with hyperphotographic memory remembers everything, every little detail. The wind direction, the sun's location in the sky, the tone of your voice. One woman described it as follows: rifling through her memory is similar to viewing little videos moving simultaneously. When somebody gives her a date or a year or something, she sees all the little movie montages on a time continuum. Now, personally, I think this would be one of the most awesome things to have. I would love to have hyperphotographic memory myself.
Alright, so coming back here, those were some pretty cool mutations that were actually beneficial. So not all mutations are harmful because, you can have the mutation where you're resistant to the bubonic plague, or, where you get super jacked because you have, that extra sequence there within that gene. So first, we're going to look at what a mutation is, and we probably already talked about it, before in another science class. But a mutation is a change in the gene sequence which sometimes results in a phenotypic change. So that gene sequence could either be like a gene mutation or even a chromosomal mutation, which we'll get to in a second. But notice that sometimes it results in phenotypic change. Not all mutations are going to show in your phenotype. They're not all going to make a physical change. They can just stay in the genotype there. Now, moving on to the next one here. So factors that affect mutations. There's a couple different ones. You got environment, hereditary, mistaken DNA replication. So the environment, let's take a look at the environment. You have a mutation within the environment because you may need to survive in that environment. And we'll actually have a picture later on in the video here to show that. But sometimes organisms, natural selection occurs, they have a mutation because of the environment, and for them to survive, they have to have that mutation. Hereditary, we talked about parents. Parents can give you different types of mutations. Notice, remember in that video that it said for that bubonic plague that both parents had to have that mutation for the kid to get it as well. So that's an example of your parents passing along a mutation. Mistaken DNA replication, during DNA replication, we've been seeing, all these nucleotide changes, and then that DNA going from RNA, making those proteins. So this DNA replication, those mistakes, can actually happen. Even though, remember in that cell cycle where we talked about that G2 phase where the G2 checks for mistakes, they still can actually occur within DNA replication. Not all those mistakes are going to be caught and fixed there. And then moving on to the next part here. So the different types of mutations, you have gene mutations, which is a mutation in the gene sequence that makes up a gene. This is a gene mutation right here. One eye is blue, one eye is kind of like normal yellowish type. Or a chromosomal mutation in the chromosome. Those, for example, is, like Down syndrome, those type of mutations, because it's a change in the chromosomal sequence there. And then going to when would it actually affect the phenotype? So mutations, like we said before, do not always affect the phenotype. They do affect the phenotype, though, when the mutation enters the amino acid sequence of a protein. So, DNA replication, we got transcription, translation. Now, if that mutation actually occurs when, you have that mRNA and tRNA, and it has an amino acid and it affects that amino acid sequence, that protein is then going to be wrong. So that's when those mutations there would actually affect the phenotype. But like I said, that's not always going to occur. Sometimes it's just the genotype that takes that mutation. And then this is the picture here I was talking about earlier. So all mutations are not harmful. We kind of tend to think that, if you have a mutation, it's a bad thing. Not always. Look at this mutation here. We have this bird with the long beak and this bird with the short beak. And notice that mutation is actually beneficial to this bird because it can reach into the wood and get those slugs that are in there. Whereas if it didn't have the mutation, it would not be able to eat. Eventually, it would die. So not all mutations are bad. Some can actually make proteins that will actually help the animals there. And then we're moving on to the left-hand page here. You need to know these different types of mutations. So the first one we're looking at is an insertion mutation. Notice you have A, B, C, D, E, F. Now look at this mutation, A, B, C, D, L, E, F. So what an insertion is, and it kind of just like it sounds, it's the addition of one or more nucleotides into a DNA sequence. So instead of looking like this, where it should be A through F, it now adds an L in there. So that's an insertion. So when you add one or more into a DNA sequence, that's an insertion mutation. Move on to the next one here, a deletion. Same thing, sounds like it is. It's part of a chromosome sequence of DNA lost during replication. So notice you have A, B, C, D, E, F, and then you have A, B, D, E, F. So notice that C nucleotide got deleted. So that's a deletion mutation. Now, duplication. Duplication is when a chromosome gets an extra copy of itself. So notice A, B, C, D, E, F, same thing. A, B, C, D, D, E, F. So that nucleotide with the letter D duplicated itself, and instead of only being there once, it's now twice. So that's duplication there. Inversion. Alright, so inversion is a segment of chromosomes is reversed end to end. So notice A, B, C, D, E, F, the normal, and then when it replicates, A, B, E, D, C, F. So those were inverted. So the E and the C were inverted, and then just the E and the C were inverted because you got A, B, C, D, E, F. So it should have read A, B, C, D, E, F, but instead it's A, B, E, D, C, F. So that's the inversion mutation. Now, translocation here is that last one you need to know. Translocation is when part of a chromosome is relocated. So look at here, you got A, B, C, D, E, F, L, M, N, O, P, Q. Now, after replication, it's A, B, C, O, P, Q, L, M, N, D, E, F. So that D, E, F up here now translocated down here. And that O, P, Q down here now translocated up there. So that's translocation, when that part of a chromosome is relocated. And that's the end to our video here on mutations. Mr. signing out.